scientific research, yeah. And he works in the laboratory in oxidative stress and disease. He has his educations from institutions in Hungary. And his laboratory is NIH funded, and they're interested in the in vivo role of free radicals in reactive oxygen nitrogen species, with an emphasis on diabetes, insulin resistance, and diabetic neuropathy. So again, his laboratory is interested in focus on pathologies that increase free radicals, including the reactive oxygen species. And these molecules are generated in normal metabolism, as he'll explain, but under healthy conditions, their levels are controlled. And of course, when we're talking about diabetes, we're talking about another state that other than health. So I'm going to turn it over to you to tell your story.
Thank you. It's good to be back. I don't know how many times we did this, but you guys always change. And, I already know a couple of you, and you may have seen the whip. It's going to be different than the whip, so you're not going to fall asleep. I don't know why we do this at 3:15. It's kind of like my brain is already, it's not catching. So what I usually do is I keep it as short as I can. And the first time I heard about free radicals was probably not until my PhD studies, and then these obscure detection methods that I do, like electron spin resonance spectroscopy, I never heard of before I get to my PhD. So I give a little bit of background on what is a free radical, what do we exactly call oxidative stress, because people float these things around in the literature and it's not necessarily always rigorously addressed. And how do we detect them? What Carol talked about is what is the balance between signaling and disease pathology. And then I'll delve into a little bit of our research, which is diabetic kidney disease and nephropathy, and what exactly radicals do in nephropathies and how they contribute to diabetic complications. So first I'll talk about what is the old school approach to oxidative stress and what is the new school, how do we define redox sensitive signaling, how do we detect these radicals such as electron spin resonance spectroscopy, which was my training in biophysics, and what is immunospin trapping. I'll talk a little bit about my pet peeves that are companies and kits that they sell to detect oxidative stress and yet they don't detect anything, and which one is to stay away from and which one is good. And then, like I said, I'll go a little bit into my own research of what exactly redox signaling is doing in diabetic kidney disease. So the definition is that free radicals that we often hear about, they're atoms or molecules or molecular fragments with a unpaired electron or unpaired spin on it. So I give a couple of examples here, right? Like superoxide, hydroxy radical, nitric oxide, lipid radicals, tyrosine radicals. So by definition, because they have a free electron, they live really short lives and they're really reactive. Because you don't want an electron to be free there, you want to pair it with somebody. So the old school was initially that everybody thought that they're deleterious because they'll react so fast with your proteins, with your lipids, with your DNA. God forbid that it's just deleterious and it's not good. Then it's been known now about 20, 30 years that just because you have a radical, that's fine, but what are those radicals doing there? They also play important roles in ambient cellular signaling mechanisms, and I'll bring a couple of examples of that. So it's always a redox balance and imbalance, right? In a disease, either the antioxidant defense will get thrown off or the radical is going to get overproduced, one or the other. And in a balanced state, in a healthy state, they'll drive important signaling mechanisms. So for the disease states, there are a couple of examples, such as lipid peroxidation, which we hear about a lot in disease models. Inflammation can drive oxidative stress through upregulating INRs and NF-kappa B. Ischemia reperfusion is a problem in organ transplant. When they take out the organ and put in the new organ, it's not the ischemia that causes the radical production, it's the reperfusion. When the oxygen burst comes in, produces superoxide, and to prevent organ death is kind of a troublesome and problematic area. And there's a lot of mitochondrial diseases. The mitochondria is a powerhouse of energy production, and it's also a powerhouse of free radicals because it always leaks a little bit through the membrane. So there's a lot of focus lately on mitochondrial diseases, including diabetic complications and excess superoxide and oxygen radical formation from the electron transport chain. And then there's the famous examples of when they're beneficial. When we're talking about redox-sensitive signaling, and I always bring up nitric oxide as a Nobel Prize radical because that was an accident, right? So Viagra was in trials for, I think, cardiovascular disease when somebody discovered, patients came in and they said, it's working. What happened to me? It was nitric oxide that does vasodilation, so nitric oxide became the famous Nobel Prize radical. So we do need nitric oxide, just don't need too much. There's also a lot of emphasis lately on protein tile signaling. So anytime you have a protein tile with an SH, it's going to be able to pick up a radical, like a cysteine. It's going to be able to pick up a radical and make an SS bond, and that's how the signal gets transduced. So again, that's a homeostatic signaling mechanism. And you will see examples in podocytes in diabetic kidney disease versus normal kidney, how that signaling goes. And then there's the opposite of vasodilation, with H2O2 or superoxide actually mediates vasoconstriction. So that's another physiological example that I brought when radicals are necessary. So there's a gold standard, and this was my original training in biophysics, how do you detect them? So like I said, the problem is they're short-lived, so it's really hard to catch them. What would be a good detection method, not just to detect the fingerprints of radicals, but the radical itself? So it's called electron spin or electron paramagnetic resonance spectroscopy. So first it looks like a totem, but once somebody explained it to me on my PhD how it works, then it became really clear. So what you see is, I have one of these in my lab, 2 magnets, and then a microwave bridge that gives a microwave frequency. So what you need to think about is your microwave oven. When you put in the food, the water starts spinning because the water is dipolar. And that heats up your food. So what happens here is the same thing, except the microwave oven is 2.5 gigahertz, this is 9.5 gigahertz. So what will happen is you put in your sample between the magnets, tissue, cell culture, or an in vitro tube, or whatever you want, and your radicals will line up with the magnetic field. So that's what I show here, right? So one would be one would be north and one would be south, right? Your spins will line up either this way or this way. So when you put in a microwave frequency, they're going to start oscillating because they're going to absorb the energy from the microwave. So what you detect basically when your spins flip. So let's say they line up this way, they all flip. How many radicals you have, it depends on how big of a signal you get, and what kind of radicals you have, it depends where you get your signal. That's kind of how you can detect free radicals, and that's the gold standard detection. So again, it's an energy split that's caused by a magnetic field, and then the absorption, it's like any other spectroscopy. Instead of light, the absorption is microwave. It flips your spins around. So that's all nice and handy. What you see is, so here's the absorbance. For some reason, the convention is that we don't show the absorbance, we show the first derivative, which is a peak. And again, like the more radicals you have, the bigger peak you have. And then it depends on what kind of radicals you have, you'll see splits in the signal and the magnetic field. So the advantage of it is, if there's no radical, there's no spectrum, so there's no guessing, like in the fingerprint method. Well, it may be, it's a fingerprint from a nitric oxide. No, if there's no radical, there's no signal. The disadvantage is exactly their lifetime. It's often nanoseconds or milliseconds. So by the time you put the sample in between the magnet, they're gone. So what you're going to do about that? I'll show you a couple of tricks for that. Relatively low sensitivity, so it never really became too popular in vivo because the sensitivity is 1 micromole, and often we don't have that many radicals in a given in vivo system, so you have to have some boost to detect them, and sometimes the spectra gets really complex to resolve. Water, like I said, it works exactly like a microwave oven, so if you have too much water, such as tissues, cell cultures, the water will spin too, and it gives a really noisy spectrum, so you have to watch for water, and there is ways around. So what are the ways around this? If they live so little, you can do what's called freeze trapping, or you can do what's called spin trapping. The freeze trapping is basically just stick your sample in liquid nitrogen, everything will freeze. And you detect the spectra, but now it's not going to decompose in milliseconds. Spin trapping was, I think, described first in Janssen, and it's nothing more than take a compound, a chemical compound, and inject it into your cell culture, your animal, whatever you're working with, and it's going to make a more stable radical. It's still going to be a radical, but it's going to be a lot more stable radical. It maybe lives hours, even days, so now you can detect your radical of interest. The good thing is that you're going to have what's called splitting constants, and there's a database, and based on the database you can look up. I'm showing a lipid radical here. Well, I have a lipid radical, I have a tyrosine radical, I have a superoxide. It will give definite splits because of the spin and magnetic quantum numbers of the given radical. Freeze trapping I already mentioned. You can also see, because they're also paramagnetic, you can see metal ions, for example, the mitochondrial chain complexes. You can see if you put your sample in liquid nitrogen. So the significance of this in biology is that you can distinguish between radicals and primary and secondary radicals, and you can understand your redox mechanism. So instead of saying we detected ROS, you can say we detected superoxide, tyrosyl, lipid radical, and whatnot. So when they developed spin trapping, there's a couple of considerations were given. So first of all, it cannot be toxic what you inject to the animal, because then you're not going to detect anything if you kill the animal. Or you get ex vivo artifacts. Cell culture is still a little bit of a trick, even nowadays, because cell culture has two problems. The oxygen is the best trap ever, so it's going to trap the radicals before you trap it. And the serum is also the best trap ever. So you have to do serum-free, but not every cell likes serum-free. So there's a limitation with time there. And it seems for some reason that for these reasons we need really high, like 10 to the 8, 10 to the 9 of cell culture, of cell number, and it's not always achievable with big cells. And it's kind of somehow like a lack of a field or expertise, or I don't know what we did, but we still don't have a good method for intracellular superoxide. Like you hear a lot about superoxide. What we can actually detect is extracellular superoxide. Like intracellular, we still don't have a good method to get in there, into the mitochondria, and see how much superoxide is actually produced. What I used to do in my postdoc, there's a couple of examples here. We did bile cannulation, as most of these spin traps are excreted into the bile. You can take your rat, cannulate it, take the bile out, and put the bile into the EPR machine. So now you don't have water, it's not going to spin, it's not going to make noise. You just have the bile and a concentrated spin trap that caught your radical. The problem is that I think it represents mostly the liver and not really the rest of the body. With deep tissue extraction, so what you do is a Folch type extraction, is a lipid extraction. If you want to detect lipid peroxidation, lipid radicals, you can take your kidney, liver, spleen, whatever you have, extract everything into the organic phase. So again, no water, no problem, no noise. And you can put your chloroform extract in a quartz cuvette, put that into the machine. So now you kind of have I call it ex vivo, in vivo detection, in vivo trapping and ex vivo detection of radicals from your organ of interest. Let's say it's the brain, you can extract the brain. There's some cheap, so the machine is $250,000. Not everybody has the biophysics expertise to do it. They have these benchtop machines circulating around that is really good for the food industry, and I think somebody at LSU has it. It's not really sensitive enough to measure anything in vivo. And then we have what we call the GDOC, the Carpentier radical. You can actually generate radicals on the spin traps themselves, which is going to be a problem because it's not the radical that you detected in vivo. So what's next? Immunospin trapping. So Ron, my mentor, said, okay, but not everybody has biophysics expertise, and it's boring, and not everybody has $250,000. So what could we do for the field to help investigators detecting these radicals? And he said, well, maybe we could combine the sensitivity of immunology, like Western blots and ELISA, with the specificity of this spin trapping, and do something, an antibody that could be commercialized and sold So now other investigators would have the opportunity to detect radicals as specifically as we did. So what he developed is called immunospin trapping, and then he sold that into Axora, which is now Enzo Life Sciences or something like that. So he took a spin trap, which is called DMPO, it's a nitron, react with a radical on the double bond, makes the radical a dot, and like I said, they're still short-lived. And then disproportionates and then makes a nitron a dot that doesn't have the dot anymore. So if it doesn't have the dot, now you missed your window of opportunity with EPR to detect it. And then he said, but look, this structure is unknown to nature. It doesn't exist in an animal. So if it doesn't exist in an animal, we can make an antibody against it. So he took this nitron a dot, stuck it on a carrier protein, put it into the rabbit, and that's how the anti-DMPO antibody was born. And now it's commercially available if anybody, any of you are interested in it. So now we combine a lot more sensitive method, which is immunology, with the specificity of spin trapping. And what we can do now with the antibodies, we can see where is the radical in the organ. We can see if it is in the mitochondria. And we can co-localize it with other things in a fluorescent microscope to see what's going on. Where is the radical produced? So I think he patented it and he sold it. And when I came to the lab, okay, yeah, so here's the unknown to nature. We made an antibody against it. We took it to in vivo. So we injected the MPO into the mice of whichever disease model of interest you have or toxicology. Take your organ, fix it, cryocut it, and then we put the anti-DMPO antibody on it and a fluorescent secondary antibody on it. And now we can detect protein damage, oxidation, and whatnot with Western blot ELISA or confocal microscopy in a given disease model. And now you can localize for the first time. It will light up. Where is your radical in the mitochondria, in the nucleus, in the membrane, whatever. He also did some mass spectrometry because the protein DMPO bond is mostly tyrosine bond. It will be detectable in the spectrometry. So if you have a protein of interest in a signaling mechanism, now you can see that protein has the radical on it. I know it sounds a little obscure at first, but it's because it's so specific and not really anybody else doing this. I like to give a little bit of okay, so this is not really anything, totem. You can get this commercially and measure your radical of interest. So that's my pet peeve slide. I've seen so much in the literature that I try to bring up every second year for you guys to stay away from. Because I know that there's a lot in any given field in the literature we measure ROS, which is fine to talk about ROS when you say we have an interest in, let's say, diabetes, kidney disease, and ROS. But when you measure, state what you measure instead of just saying ROS, because it would be like saying fatty acid instead of saying palmitate. You have to define specifically what you're measuring. The second is everybody took advantage of, all companies make these fluorescent probes. Namely, DCFDH and DAG, hydroxyathidium. And they say that's specific for all ROS, that's specific for superoxide, and it's not. I'll show you why. Sorry, so if you can stay away from that. And then another interesting phenomenon is we will cure everything with antioxidants. We just give antioxidants and they will cure everything. And they all failed. Vitamin E failed in diabetes, and why exactly this happened? I'd like to show a little bit of that. So here is this famous fluorescent probes. Just don't do it. This is why you don't do it. It's a little bit of biochemistry, I know, so just bear with me. You have superoxide becomes H2O2 because it gets dismutase by superoxide dismutase, and that H2O2 will react with cytochrome C, and you need this reaction for the fluorescent probe to work. Makes the cytochrome C compound 1. Here is the fluorescent probe that the company sold. It becomes fluorescent, and you measure ROS, whatever is fluorescing. The problem is that there's a competition for this reaction between the probe and glutathione and ascorbate and NADH. So now, if you have more glutathione or less glutathione, you will have more or less compound available for the fluorescent reaction. So you either really have more ROS, or you just have less competition. You don't know if you really had more ROS, or you just had less ascorbate, which is the case in diabetes. They have less ascorbate and less GSH, so everybody thinks we detected more ROS. Maybe, maybe not. The DAG is a little bit better. It makes two products. With superoxide, it makes the ethidium cation, and it gets auto-oxidized and makes the 2-hydroxyethidium, sorry, the other way around. It makes the 2-hydroxyethidium and it gets auto-oxidized, makes the ethidium cation. The unfortunate thing is that this is 80% artifact and the real reaction is 20%. And everybody detects a red fluorescence, which is 20 nanometers apart. You can't see that in a microscope. Everything will be just red. What you can do, separate these two peaks on HPLC and see a real reaction on the artifact. So if you have HPLC, it's okay, otherwise it gets really tricky what you detect. And then, okay, so the antioxidant therapies. We think they failed because, first of all, they failed to reach sufficient concentration in the target tissues, and nobody really measures it. Like if you think about vitamin C, what you get, you basically pee it out. If you get other antioxidants or botanicals, you would have to eat 15 kilograms of them to get a competition in vivo, because Mother Nature gave you GSH and Mother Nature gave you ascorbate as antioxidant defense. So we never really measured how much of these antioxidants get, in the blood or in the tissue. So the kinetics and the pharmacological characteristics have never been considered. If you give different compounds, they have different reactivity towards radicals. So like they're not going to catch superoxide and nitric oxide the same way because these are distinct reactions. And then, like I said, it's hard to compete with what Mother Nature gave you. If you have enough SOD and if you have enough glutathione or ascorbate, you'll take care of the radicals. Not, like you mentioned, in a disease like diabetes, these antioxidant defense mechanisms may be down, so that's why you have more radicals. But again, it's going to be really hard unless we know the kinetics of these antioxidants. It's going to be really hard to cure everything with antioxidants, such as the resveratrol, unless these radicals control a signaling mechanism. And then you block that signaling mechanism, that will work. But just scavenging them won't, because you're competing again with reactions that go 10 to the 9 in your body. Good luck competing with something that reacts at 10 to the 9 and just annihilates everything. So if you don't have EPR and you don't have this antibody, you can still measure good things such as isoprostanes, before hydroxynonenal. I think a lot of you, even in the clinic, like you hear about isoprostanes. These are a really good measure of lipid peroxidation. They did it in marathon runners, in the urine, and I don't know whatnot. It correlates really well with the amount of damage that you would get in oxidative stress. Nitrotyrosine, I think you hear about that a lot too. For nitration of proteins, it's a good measure, not only for peroxynitrite. The fluorescent probes are okay. Again, I'm not necessarily advocating against them. If you have the right tools, you can use the fluorescent probes. And if you're lucky, if you have EPR or you collaborate with someone who has EPR, that's still the best way to go. But not everybody has the biophysics background to interpret the spectrum. So it's even like anything in life. It's a give or take which way you go. So we get to the diabetic complications now, and I'll give you a couple of examples what we could do, and then we'll get to the kidney disease. That is probably more interesting part. So like I said, you can take your tissue of interest in a given disease and stick it in liquid nitrogen. You're not going to see distinct radicals. You're going to see what we call like an envelope signal, like so. It will contain a lot of different radicals. But if you see an increase, then you have a good idea about your steady state. It's going up or down. Or in a disease, you have, let's say, 5 times more ROS now, but I cannot really still tell what kind of ROS. When you do spin trapping, and we did this in diabetic rats with a spin trap called POBN, that's kind of specific for lipid radicals. Now, lipid radicals could be anything from lipid peroxidation. So the LOO dot that we show, we don't know what the L is. It could be any kind of lipid, but we kind of have a good idea at least that, okay, there's more lipid peroxidation going on or not. So what we did here is we injected rats, waited an hour, took the liver or whatever organ, performed that Folch extraction that I mentioned with chloroform and methanol, and stick that extract into the machine, and then we see the 6th line. Okay, if they're bigger, then we have more lipid peroxidation. You can do the same thing with the bile cannulation that I mentioned. So here's a victim giving bile to that little cannule, and then he received a spin trap an hour before and just keep giving bath for the whole afternoon, and then we put that in a machine, see lipid peroxides. Now, if you use inhibitors as a pretreatment to inhibit enzymes, then you can go mechanistic. So what produced these lipid radicals? Well, let's systematically block enzymes that we think produce lipid radicals, such as, I don't know, NADH, NADPH oxidase, or catalase, or macrophages, and we'll see if the signal goes up and down. So now you kind of go mechanistic in vivo and see which one was your. And I think in the next slide we did that. Yeah, so we had diabetic or obese, big poofy rats and control rats, and we pre-treated them with 1400W, which is an INOS inhibitor. INOS is always associated with chronic low-grade inflammation, so that's why we were thinking about it as a candidate mechanism. And now you just compare, what we compare is the peak heights of the EPR spectra. You see how much more they have in diabetes. And then if we block INOS, it goes down. So we thought, okay, so this is an INOS, inducible nitric oxide synthase-driven lipid peroxidation. So now you can get a little bit more mechanistic in vivo, which is often tricky, and you gotta go to cell culture. And in the third example that I brought, this is with Jason and Susan Collier. They're interested in pancreas and pancreatic beta cells and in diabetes, and how they fail upon chronic inflammation, which they model with interleukin-1 beta treatment and culture beta cells. And Jason thought that there's a lot of nitric oxide produced, and that's a theory in the pancreatic field, that the nitric oxide eventually will kill the cell. And that's why you get type 1 diabetes. So we took his cultured beta cells, either nothing or put interleukin-1 beta, and we put another spin trap called MGD iron on them. And you see a 3-line spectra, which is typical for NO, so that's nitric oxide. And then when he blocks the production by nitric oxide synthase inhibitors in different doses, then you see how much your spectra gets smaller and smaller. So Jason was really happy because nobody else showed nitric oxide directly in a pancreatic beta cell. So this was the first time that him and I did active nitric oxide. So we confirmed that pancreatic beta cell killing by interleukins is probably through nitric oxide production. And if we block that, the cells live happily, and then we'll see that there's less and less nitric oxide detected. I think, okay, and I came back to that immunospin trapping finally in the kidney that Ron and I developed back then. So this is an example of when I said you can inject the DMPO, take your kidney, cryocut, and use that antibody instead of the EPR machine, and use a secondary antibody, and now you can localize where is your damage. So we fed mice high-fat diet for 12 weeks and 16 weeks, and at 16 weeks we see these green dots showing up in the glomeruli, so those are protein radicals that are detected by the anti-DMPO antibody and just visualized by a green fluorescent secondary antibody. And then we went further later, I just didn't want to get into more details. So now you can co-localize it with a mitochondrial marker, for example, see if they're in the mitochondria. So get a little bit more information than just ROS, where they're produced and what are they doing in the disease. Okay, so the second part is a little bit about my research and a little bit more on the signaling side. So what these radicals are doing and how they get to this whole kidney disease phenomenon. So I got to Pennington, it's been about 7 years now, or 8. We were talking with Jackie and a couple of others. So where's the white spot? Where could I go in where there's not much known yet? And so we decided that diabetic kidney disease and redox and oxidative stress is kind of a vague. So everybody talks about oxidative stress as a common denominator of nephropathy, but nobody really knows what's going on. Maybe with your expertise, Chris, maybe you could go into this and see directly. So that's why we picked diabetic nephropathy or chronic kidney disease. The problem is that it says it all. Once you have it, you're done. There's no cure for acute kidney injury or chronic. You probably slower or later you're going to advance to dialysis. If you advance to dialysis, this is like a 5-year ago cost estimate, which is $12 billion for the States per year. So it's tremendous. So everybody's looking for some sort of at least a prevention or postponing the complication. So you can have many, many years of diabetes walking around and you have kidney disease and you don't know it. If we could postpone it at least a little bit, well, that would be something. So what the little diagram shows is that diabetes affects the kidney. Here's a healthy filtration, right? So your glomerulus filtrates blood, makes urine. In diabetes, the protein leaks out. So then the first sign is you have albuminuria. You don't want a protein to leak out, because that means it's an unhealthy filtration unit. And so the Kaplan-Meier curve on the right shows how much of a survival you can expect depending on how much albuminuria you have. So when you have non-proteinuria kidney disease, you're doing pretty well. The more proteinuria you have, the more damage you have in your filtration system. Then the survival goes really quickly down. So it's an important problem, and everybody's on it to try to at least prevent it, catch something. So this is because none of you guys are in the kidney field. This is a single unit it's a single filtration unit in the kidney, which consists of, the blood comes in here, right? And the arterial eye goes into the capillaries. This is the glomerulus. The green cells are the podocytes that I will talk about a lot. They kind of sit there. I always show this. Podo means foot. So they sit there with their foot like a zip, and they don't let the protein out. If you have disease, they do this, and now your protein is going out. So their foot process is damaged. And we'll talk about it a lot later, because this podocyte detachment, apoptosis, and podocyte loss is usually the early event, even in obesity. When you don't have diabetes and hyperglycemia, you can still have podocyte damage. So you're happily walking around not knowing that you have kidney disease. And then once you're done with the filtration, it goes into the proximal tubulus, then comes around to the distal tubulus where the sodium uptake is done, and then it goes out. So here is a TEM and electron microscopy picture of those podocytes that I showed. So the podocyte is actually, so this is the capillary, this is the blood. The podocyte is a big cell, and a lot of little things that you see is the food process, which is like a zipper. And you see a picture here from one of my high-fat diet mice. The zipper is gone. What you see is like really flat, well, a couple of them. There's a really flat foot, which is called food process effacement. So now you're losing your zipper mechanism that holds the protein in and lets the filtrate out. So now everything is going out. And those cells are probably like apoptotic or detaching and dying, and that's why you're not having the perfect zip there. So like I said, the people were kind of fishing around this oxidative stress as a common denominator in the pathogenesis of diabetic nephropathy, and they thought, well, there must be mitochondrial sources, because the mitochondria is a powerhouse of energy production and ATP, as well as superoxide, and there must be cytosolic sources. So a little bit about the mitochondria, if you're not in the mitochondria field. Electrons get passed right from complex 1 to complex 5, because that's how the redox potential is set up. The substrates coming in from the TCA cycle, it could be pyruvate, which will feed through complex 1, or fatty acid-driven or lipid-driven substrates feed on complex 2, pass the electrons from NADH and FADH to coenzyme Q and all, go out to the intermembrane space, come back on complex 5, and you make ATP. And the process always have a little bit of a leak, and that's what I show here on complex 3 and complex 1. It's always a little bit of superoxide coming out. It's just how the membrane is leaking. It doesn't really depend on the membrane potential or anything like that, it's just leaking. But you have defense mechanisms. The superoxide dismutase and the catalase, they'll get rid of it, and then it's all fine. So what happens in diabetes is, type 2 diabetes, I would say that there's a substrate overload, because you're obese, you may be sedentary, you're sitting on a couch, so there's no ATP need. You don't make ATP, you're just sitting around. But you have a lot of substrate coming in, glucose and pyruvate and fatty acids, so they overload the mitochondria. So now what happens is you used up all your reductants, the electron has nowhere to go other than backwards. And now you're going to have a lot more superoxide coming out. So that's what we're thinking right now, and that's one of my projects. So that's where the mitochondrial loss is coming. Cytosolic source is usually NADPH oxidase as the root of all evil, and specifically in the kidney it's NOx4. NADPH oxidase always makes superoxide. Diabetes, it gets dysregulated and will contribute to superoxide production. So while everybody was focusing on the O2.- and H2O2, I took a look and I said, you know, what I talked about in the first part, superoxide is so short-lived and so reactive. How could that be that makes complications? Like it's not going to even get out of the mitochondria other than doing local damage. So it never made really sense to me why a primary radical who is so reactive would make such a far-reach damage. What if it's secondary radicals? What if they can move more? So I put my focus on secondary radicals. That's why I talked about lipid peroxides a little bit at the beginning. Because when you compare a superoxide half-life and a lipid radical, one is 10,000 times more than the other. So that makes sense that maybe could reach a little bit further than a superoxide. Superoxide is charged, so it's never going to get out of the mitochondria because a charged species cannot go through membranes. It's just biophysically impossible. So it maybe do local signaling. Okay, fine. The lipid radical now have a lipid on it and able to cross the membranes and fuse with the membranes. So maybe it can have far-reaching signaling effects, and maybe it can always do cumulative damage throughout the days and years and whatnot, because it's just slowly reacting and accumulating, whereas the superoxide gets either eliminated or makes local signaling. So I turned to these. I got interested in these secondary radicals and lipid radicals, given their lifetime and given that they can really do cumulative damage. So the way they produce this, you need an initiator that is high enough in redox potential that can initiate lipid peroxidation. And it's usually the hydroxy radical. Reacts with an unsaturated lipid. If you think about the mitochondria, forgot to tell this, it's a lipid-rich structure. So here is superoxide produced right on the ETC. Here is the lipid. I bet it's going to attack because it's so close. And there's not many other structures in the cell that are like that. There's a powerhouse producing radicals, and there's a lipid structure close. So I bet that in diabetes that would increase lipid peroxidation in the membrane. So then you have a lipid radical. It's either going to react with itself and then makes a lipid peroxide, or reacts with oxygen, makes a lipid peroxy radical, and then again the end is lipid peroxide. So there's a lot of interest in them lately because it turns out that, well, these are not just deleterious either. They can drive signaling mechanisms. And that's what I'm trying to show here, that you need a little bit, like you always need a little bit of everything. If it's a lot, that's a problem, but they're electrophiles. So they can react with nucleophiles. That's basic biochemistry. What is a nucleophile? It's your protein, cysteine, histidine, and lysine amino acid residues. Specifically, the cysteine is really keen to react with electrophiles, such as these electrophile lipids. So there's a lot lately in the literature about reactive lipid-driven redox signaling, and when it's deleterious and when it's not. So here are a couple of examples of lipid hyperperoxides and reactive lipids, acrolein 4-hydroxynone and isoprosteins you guys probably heard about. So if you're in a low range here, then you're driving signaling mechanisms through the cysteine protein tiles. If you have too much, now nothing is specific when you have too much. They're just going to hit everything and make protein carbonyls and unspecific reactions. So then we're talking about oxidative stress. But I think it's easy to see that when you have a little bit, that you can have targeted mechanisms, because the cysteine will be the first one close to you that's going to react. It's not specific, but it's just because he has the best redox potential for it. He'll go for it. And then if you have a lot of lipid radicals, everything will go for it. So when I got into this, we did a screen in what is a protein that is a major regulator of cell fate and life and death and motility in the podocyte in the kidney, or in other cells too, and it's redox sensitive. So we ended up with RhoA. What's specific about RhoA is that RhoA has 2 cysteine residues, shown here, and he's a master regulator of cell fate and death and life and motility. So you want Rho A to be active, but just active enough that your cell will do the proper cytoskeletal structures. And if you have too much Rho A or too little Rho A activity, that's detrimental to the cell. So we ended up with Rho A as a master regulator because of this. And then this paper came out that says, well, Rho A can be activated by ROS. Well, fine. Because it has 2 cysteine residues, and like I said, that's the most reactive towards radicals. So maybe this will control cytoskeletal distribution and survival. Nobody knows in the podocytes what does that. Yeah, again, so it's a master regulator. Turns out from papers it's a master regulator of podocyte motility in the kidney and F-active arrangement. You have an inactive form that is GDP associated. The GDP has to dissociate upon signaling or ROS attack or whatever, and your Rho A gets GTP on it when it gets active. And then it moves the cell, or doesn't move the cell, or moves the cytoskeleton around. So it's going to be really important for podocytes because podocytes want to do this food process. If they can't make the food process, everything will fall apart. And Rho A is the one who controls the cytoskeletal motility. Cytoskeleton is the one who makes the food process possible. So I started to see the connections there. So we said, well, we're going to take this. So we get immortalized, conditionally immortalized podocytes, which they come from the immortal mouse. You either wait 6 months for the immortal mouse, or you just get it from your bodies. So we get the cells from Houston. You can passage them at 33. If you put them in 37, they differentiate. And then you do your experiments, and then you can throw them away. Because they become conditionally immortalized but differentiated podocytes. And they express what's called synaptopodin, that's characteristic, that they're differentiated. And then we said, well, we know all this about radicals. We cannot only scavenge radicals, we can also generate radicals. So why don't we generate lipid radicals in vitro, throw them in the podocyte and see what happens. Do they move? Do they not move? What are they doing with their food process? So AAPH is a chemical compound. We put them in the incubator with this AAPH and will generate alkyl radicals. They form alkyl peroxide radicals with oxygen. It's just like an in vivo lipid peroxidation, except I can control it. So how cool that is. There's this nice equation that the guy gave in the paper. So how much AAPH you put on your cells with this equation, you can calculate how much radical you generate per hour. So let's say with 10 millimole, we generate this much per minute or per hour. With 25, we generate this much. So we could do a dose-dependent test of how this redox signaling will really go in the kidney. Well, at least in vitro. So again, the theory was that you need a little bit of lipid peroxide to activate the cysteine residues, and that will provide a basal Rho A activity to control the food process. And that's a healthy kidney. Now in diabetes, if you have too much lipid peroxides, the theory was that it's going to be either an overly active Rho A, and then the cell's not going to move, and not going to be able to make the zip. So now you die, and then you have all these proteinuria. Or it will make a cysteine-cysteine bond, and then again, it's not good because now you have hypermotility, and when you have hypermotility, everybody's going to fall off the basement membrane, even, instead of doing this, the zip mechanism. So either or. So when we did the first experiments, the lipid radicals do or do not activate Rho A. We did an ELISA, and yeah, they do activate Rho A in a dose-dependent fashion. So I said, okay, so this is going to work. Instead of just ROS or superoxide, lipid radicals activate Rho A too. So we took our podocytes, and this is kind of an in vitro surrogate measurement for a food process. They don't really have a food process in vitro like you would see in the kidney, but what you can do is you can culture them and then scratch them with a pipette tip, and then you wait 3 days and look at them. They're going to migrate in. So it's not really food process, but it's migration, so it's kind of a surrogate measurement for what in vivo would happen. And we throw different amounts of radicals in them, and then you see that all of a sudden at 2 micromolar they don't migrate anymore as fast as they used to. I said that Rho A will control how they make their cytoskeleton, and that could be measured with an F-actin staining, how healthy is your cytoskeleton. So what you see here is that they make these, it's called stress fibers, but it's not really stress. They make these very organized cytoskeletal structure, and if you throw radicals on them, they kind of make these actin aggregates that are the dots here, the little dots, and you don't see the green stretch fibers anymore. And then with the ImageJ program, you can measure what's called anisotropy, how parallel the cytoskeletal fibers are, and they're not parallel anymore. So you kind of everything is falling apart. Let's see how this works. Because Michael Saldom came to me and he said, I cannot go back there. Because the mouse wasn't working. So I did a movie on a microscope and just watch and see. This is a 3-day movie. You see the control cells on the left kind of migrating into the wound, right? And then you see the cells treated with radicals just kind of sitting. Just watch the front. See, they're already coming in from the other side of the scratch. And here it's like, nothing really happened. So Michael said, why don't you make a time-lapse movie and get a lot more information out of your migration assay? Like, how do they really migrate? And once you're done with this, then what you can do is get an ImageJ tracking plugin and track 50 of these cells or 60 of these cells, how much they migrate, and then you can measure the distance, you can measure the velocity, because you know every 5 minutes or every 30 minutes we took a pixel. So counting the pixel numbers, you know how fast they're moving. You can calculate velocity over time, and you can see their direction. So what is really going on when they get lipid radicals in them? So you see that the total distance and the average velocity is a little bit less. It's significant, but it's not like wow. But when you look at the little colorful, like sausages, I tracked a couple of cells there. You see that they move forward if they don't have radicals. And again, think about the in vivo. Move forward means I'm going to make my foot, and I'm going to have the zipper. And what you see with the 2 micromolar radical dose, that they kind of just... So the distance that they make is still a distance, but they're not moving forward. They just kind of, they lost their directional control. So now you're not going to make that zipper foot. So why is that? So when you look at a podocyte, row A is at the tail end, and row A always have other migratory protein partners, which is CDC42 and RAC1, they're at the front end. The convention is if row A is down, the others are up. How is it? And the others are down and row A is up. So in principle, when row A is down, just a little bit active, and RAC and CDC42 are more active, then you're moving forward. So if you remember, radicals activated row A. So we think what happened is that when lipid radicals activate row A through the cysteine residues, but then these guys are going to go down. So instead of moving forward, you saw those kind of just sitting in one space, circular movement. Row A kind of pins the cell down. So imagine just, now somebody's holding your tail and it's kind of, you're just sitting there and you can't move forward. So I think that's probably what's happened. And then there came Jason again, and Susan. I said, Chris, we can mutate the cysteines for you. This is what we did in our whole life. So I'm like, how cool is this? Because the only way to figure out if this is really a redox reaction is to take the cysteine off, mutate it. Your row A is still going to be healthy row A, but it's not going to respond. So not going to respond on any radical treatment because the cysteines are missing. The way Jason and Susan does it, they mutate it to alanine, and then we put it back in the cell. So now we're going to have this dominant row A there with alanines. If you activate row A with other factors, it will still activate, but not with radicals. So now we're decoupling redox sensitivity from its activity. So I can answer, this is just in my head, or this is really the mechanism. How are they moving forward? Are they moving forward because of lipid radicals are too much, they got pinned down or not? So they did the constructs. This is just to verify that I have the right constructs, and they get into the podocytes. Dose dependently, they did the GFP tag on them, and so you can tag them with GFP, that they're really in the cells. It looks much better here than there, I swear. We did our ELISA, and you don't see more activation. So radicals activate row A, when you don't have cysteine, there's no activation, okay. So now he said, we're going to put this in the cells. So now you take your cells, transfect them with this adenoviral-driven transfection, and then wait, and then put the radicals on, and then do your scratch assay. You see the normals, right? So they stop migrating. You see the wild types. So there was always a like Z kind of wild-type control, they stop migrating. You see they're double mutants, so we have two cysteines, right? So we have to mutate both probably, cysteine 16 and 20. So they did both. They migrated. They're migrating pretty well, no matter what kind of radical I'm throwing. So it looks like we got the right thing, that it's really through the cysteine residue. They can organize their cytoskeleton, right, with radicals. You mutate the cysteine, they have no problem organizing their cytoskeleton. So again, it was a redox-driven, ROA-driven mechanism through the cysteine residues. And then I think I did the same thing. Let's see if I can activate this again. Because we did not have a mouse pad, and it was like Patty was trying to copy the file. There you go. Just have to be really like kind of struck them together, otherwise I'm cheating. But you're gonna see that they both migrating the same way. So again, these are the mutants, right? So both cysteine are gone. There's no redox sensing. So they're coming too. They just started later. Yeah, so they're coming. And that's what you want, right? So if they can migrate, then they can make the ziplock mechanism. But they still migrate. So they do maintain forward track when you look at, distance and velocity. But when you look at the little colorful sausages that I tracked, you see that instead of getting pinned down, and just kind of sitting there, they do maintain the forward track. Again, it's a surrogate, but in vivo, it would mean that in your kidney, they develop their food process, so they maintain the filtration barrier. So we thought, okay, this is new and this is prominent because in diabetes, they talk about lipid peroxidation so much and these reactive lipids that this is one mechanism out of many how lipid radicals can or cannot control podocyte fate and apoptosis. And then Jason did, with Susan, they did the single mutants. So what happens if you mutate one cysteine or the other? When it looks like that's not enough. Makes sense because you have the other cysteine. So if you just mutate one cysteine 16 or the cysteine 20, they can't move forward when you treat them with radicals. So you need both cysteines for the redox mechanism to work. What's this? Oh yeah, so we're trying to take this into in vivo because that would be an ultimate nice goal. How do you generate reactive lipids in vivo is not always straightforward, so we did the kind of a trick with Randy. And we modeled this with knocking out an enzyme called carnitine acetyltransferase, or CRAT. So Randy has this flux mouse that we took advantage of, and we mated it with a podosyn-3 mouse, so they had a podocyte-specific deletion of this enzyme called CRAT. And I brought the mitochondria up again here. Because what CRAT does is, CRAT is in the intramembrane space, and it takes out preferably short-chain acyl and acetylcarnitines, which are produced here from the citric acid cycle, and they accumulate, and then CRAT takes them, shuttles them out from the mitochondria so they don't accumulate there and become toxic. Why we took this approach is, if there's no CRAT, these acyl and acetylcarnitines will accumulate, use up all the reductants, so I can generate that back pressure that I was talking about. And if I don't have CRAT, I'll have a lot more superoxide and OH, and hopefully I'll have a lot more lipid peroxidation. So it's kind of an indirect approach. How am I going to generate more reactive lipids in vivo? I cannot inject reactive lipids there, but at least mitochondrially I can get close to this. This is a rich membrane, rich structure. I don't let the products out. I'll use up all the reductants. These superoxide radicals will hit everything and generate lipid peroxides for me. So we're kind of in the baby steps with this. Yeah, so here is the explanation. Again, shortage of NADH will lead to an environment conducive to radical formation and can initiate lipid peroxidation. So this is how the phenotype looks like so far. The podocytes would sit there in the glomeruli, right? So you see that there's a lot of sclerosis going on. That's the pink material. And then there is a specific staining only for podocytes, which is called WT1. WT1 is a nuclear marker, so you just see dots. When you count the green dots, you can count how many podocytes you have as WT1 positive cells, and then in the mutant, you see you have a lot less. Again, it's detachment and apoptosis and podocyte loss to the urine. So it looks like we're creating a good model for chronic kidney disease, where you have the first sign of chronic kidney disease is podocyte loss. And then we'll have to go on and, like I said, this is the beginning, measure albuminuria in these mice as the surrogate measurement for, like, okay, do they have albuminuria or not? Do they have kidney disease? Do they have changed blood glucose, or we have to mate them with the diabetic mice to make a cross or something like that? So that's how far we get. With the in vivo model, I just throw that in there because I never showed this to anyone before. That's cool. So we get to the end of it. So what I was trying to say that, you know, there's a lot of talk about oxidative stress, there's a lot of talk about redox signaling. They definitely play a role in a lot of diseases, including diabetic nephropathy and podocyte injury, but I think it's a lot more fine-tuned than people thought before, and there's always a scale of, you don't want to just scavenge everything, because those radicals are needed. You need to go specific and targeted if you can. So you don't block the physiological response to that. You know, like I said, a little bit of ROE activation is needed, but just not too much. So how are we going to go about this? It's not easy. There's a new and exciting field, which is reactive lipid species, and they call them RLS, because people think that not only in diabetes, but in other diseases, this cysteine trigger protein tiles, which is going to be specific mechanisms where we can figure out which way the signaling is going and which way it's pathological versus adaptive or maladaptive, so to speak. Hopefully I showed you a couple of powerful and trustworthy methods to answer redox biology questions and how would we go about detecting radicals. Unfortunately, like anything else, there's never an easy way. And if you ever come across oxidative stress or ROS, just think about it. Why would you have radicals in your system? Because people often come to me, you know, from other institutes, from the clinic. And here's a piece of heart from a patient. Can you measure radicals? They're gone. But why do you think you have radicals in that disease? So think about the biochemistry. Can you have radicals in the disease, or it's just you just wanted to measure ROS because you saw it? What kind of species you have instead of just saying ROS? Am I interested in more superoxide? Tayu and Rob and I do a lot of these. Am I interested in H2O2 because they come out of the mitochondria in the muscle in insulin resistance? Am I interested in nitric oxide, and why instead of just saying ROS? And what would you like to prove? Would you like to prove that, you know, your disease pathogenesis is associated with oxidative stress and it's harmful, or would you like to prove a signaling mechanism through which, you know, nitric oxide did this famous discovery, or other radicals activate ROE and cysteine residues in my case? So always have those questions ready, you know, before you want to delve into, like, why. It happened to me so many times that clinicians come to you with a piece of heart. I'm like, you don't know how to detect this. And I'm like, they're gone. And why do you want to know? Maybe that's, you know, my mentor always asks me, why do you want to know? And if you're really interested in this, this is the website that we go, and I'm on the website committee, and this is our annual meeting. There's an annual free radical meeting with about 600 people. I tend not to go anymore because I have to go to the kidney meetings. There's no free radical biology study section where my grants go, but there's kidney study sections, so I better go to the kidney meetings. And this is Chris Cavill from Shreveport, who is actually in the search committee for the new director now on the web. So if you're really interested in radicals, then you can go to sfrbm.org and learn more about it, or just ask me if you have any questions. And that's what I have.
Time for questions.
Yeah, I was too fast. Because I tend to be too fast.
You did move pretty quickly there. So, questions?
With your lipid radicals, yeah, if you're looking at crack, why is it not also looking at the crunching acyl transferase? What, getting lipids out of the...
So yeah, so you can get lipids out, acyl carnitines out of the mitochondria if you don't have crack. So what's going to happen is that they just keep fueling that NADH, FADH electron chain, and use up everything that was available there. And they just keep coming in, so now there's no way to match the NADH and FADH to that. So I was hoping that that way I can create oxidative stress. Because now you're not shuttling the electrons to ATP. You're shuttling the electrons backwards to get them out on complex 1 and make superoxide. So that was the thinking. I'm still not sure that it's 100% perfect.
My next question is the podocytes. I mean, in the cell culture, can you tell how does that relate to in the kidney or that?
Yeah, no.
Is it activated in this direction
No.
or is it going apart? How do you know?
No, yeah, good question. Nobody was able to do food process in vitro. So we can do food process in vitro, which way they're going. So it's kind of a commonly accepted surrogate measurement that you scratch and you see that they're moving, then you assume that that's what you want. And like it's a double-edged sword. Because you don't want to move too much, because what happens in vivo, then you fall off the basement membrane and you're gone. You know, if you move too little, like the other direction, like you said, now you can't make the foot. But what if you move this way too fast, then you fall off. So no, in vitro it's a surrogate and there's no way around it. We need to look in vivo in the model, in the crack model, and take the transmission electron microscopy picture that I showed from the high effect and see if they're effaced. If they're flat, that's what I want to see. But in vitro, nobody in the field ever been able to do food process. They just don't develop food process in vitro. So that's why we look at the movement. Which way they're going? They're going that way, right? Because I made the scratch there. So there's no space. That's the only way they're going. Because if I make another scratch the other way, they would go that way too. So it's kind of you're right, it's an artificially created, I made a scratch so you better move into that space. Because I didn't give you any others, they come from.
So what's the significance of the scratch in vivo?
So the significance of the scratch assay is that at least you have an idea of if their cytoskeleton is functional or not. And then in vivo you have to look, you need to look at the TEM. So the surrogate would be, yeah, if they don't move, people always assume that that's a bad thing because then there's no food process.
So then what is that equivalent to, like in an actual animal model?
In an actual animal model, that would be equivalent to the FA stuff, that they get flat because the cytoskeleton is not working. So instead of the zip that you saw, you would see that flat. So this would be the equivalent if I could get to it. I have it. That would be the equivalent in vivo. So this is what you want to see. Right, those are all, each one of them is a foot. And then this is the flat. So in cell culture, this would mean I'm moving, and then this would mean I already fell off or I'm just sitting there. It's good that it came up because nobody ever figured this out, how to do this in vitro. And in vivo, it's so hard to do mechanistic. So they correlate the two surrogate things together, but when you take an immortalized culture, you never develop foot, ever. Nobody was able to see a foot.
Are there upstream things that occur that affect diabetic kidney disease, like circulating metabolites that?
Yeah.
So have you targeted any of those?
No, but good question. Yeah. So inflammation is one of them, and it always comes back, and infiltrating, well, not so much circulating things, but infiltrating macrophages in the kidney.
Is that what's suspected, the INOS?
Yeah, it's the INOS and also a lot of this, the fibrotic process. So forgot to mention, but this is the first thing, ultimately you end up with fibrosis. And that's the tubular cells, that's not the podocytes. And in the tubular cells, they always see this macrophage infiltration. So what they're thinking is there's molecules released, like you said, circulating or not, or from the macrophages, that trigger this TGF-beta driven fibrosis, and that's how you end up with fibrosis. So one thing would be to look at the macrophages, and is there any INOS or radical production there? And if I block it, or block the macrophages somehow, like gadolinium chloride would kill them. can you prevent that fibrotic process?
Mhm.
Then there's a lot of other, there's a lot of clotho people lately. We're looking at beta clotho, and FGF23, I think, is in the kidney. That is clotho. The clotho is protective of diabetic kidney disease, and it comes from the circulation, released from who knows where yet. So there's a lot of people on clotho and FGFs.
You had mentioned a antibody-based method for detecting radical oxygen species. I noticed that in the workflow, if you're doing it in vivo, you take rats and you inject them or feed them that molecule at a gram per kilogram per day.
Oh yeah, the DMPO. Yeah.
Wouldn't that in itself create a great deal of oxidative stress? I mean, you mentioned it's not native to the body, so I mean, it's going to create an immune response at that high of a level.
Yeah, it will. It will. So that's one of the, I think, the tests that they're doing. That every spin trap that is designed is supposed to not create that. I believe that they probably do a little bit. Or the other way around, it's also an antioxidant. So how much you want to give. You may not detect the radical that you wanted to detect because it's a scavenger. So it will catch it. But if you block the initial production of that radical, then at the end you're not detecting anything because you block the source. So those are considerations, yeah. And then I've seen mice, when we did these, mice don't take it really well at 1 gram per kilogram, and they start shaking. And what we think is either hypothermia, so there's something there that gets deactivated. Or what other groups think is that the myelin around your nerves get disintegrated with the spin trap, and they just start doing this.
Wouldn't it cause vasoconstriction?
Yeah.
I mean, your
Yeah,
body.
and that too. So yeah, so the system is not perfect. So we have to like go through a lot of these, like how much is, you want to inject a ton, otherwise you're not, so the problem with the field is that if you're not injecting a ton of these spin traps, like you said, then you're not catching anything. If you inject a ton, now you're going to interfere with immune metabolism, immunology, maybe vasoconstriction, maybe thermoregulation, so you want to be really careful like how much you give to an animal. And I think that's one thing why the EPR never really gotten too popular. And I'm probably one of like the late dinosaurs that still do it. Because it's just not sensitive enough. Because you kill the animal with the spin trap before you detect anything, right? So the drawback is that, like I said, you don't want to block everything, but you're saying is true. You give enough spin trap, you will scavenge everything that you can. But now you're blocking important mechanisms that would control, let's say, vasoconstriction or would control thermoregulation. So I think the field is heading towards like a more targeted and careful design of these spin traps. I didn't mention the mitochondrial targeted. So what they do now is they have a spin trap, will catch the radical, but they put a charge on it. Because of the charge, it will go into the mitochondria. So, but it doesn't go anywhere else. So now you can focus on your mitochondrial ROS, but without, let's say, interfering with the immune system. And then like you see more and more, I think people go a little bit far-fetched. It cures everything, and MitoQ is famous. It's a semikinome, it's mitochondrial directed, and it's been shown to cure from fatty liver disease to diabetes, everything. It's like respiratory. So if it cures everything, my first question is, what's the mechanism? There's no way it's the same mechanism in a fatty liver than in a kidney, right? So how do you cure everything is always a little bit, somebody picked this up, it's really flashy, and then 10 years later it turns out to be nothing. We've seen that before. So yeah, the spin trap design is going a little bit more the careful way, controlling for what you have.
So you talked about some of the antioxidant enzymes. What about high doses of vitamin E? Has anything been done with that, or do you
Yeah.
have any thoughts on that?
In diabetes, they failed, yeah. They did vitamin E and they failed. I think where we're failing is, again, you cannot give a high enough dose to compete with something that reacts 10 to the 9. If you look at these radicals, most of it, it's diffusion. It's 10 to the 9. By the time you inject the high dose, they already reacted. So the way around is, if you have a compound that would increase the activity, of the antioxidant enzymes. That's a hot field because you want to increase SOD activity. People never measure this. Like, so that's, I think, where the antioxidants fail. We give antioxidants and we never show the SOD activity is increasing, maybe. Those enzymes are really fast. So SOD, for example, is, I think, 10 to the 7. So now you have a shot against the 10 to the 9. If you can increase SOD activity a lot, or NOS activity a lot or something, catalase activity, people put catalase in the mitochondria, and that has been shown to increase insulin sensitivity. Because now you're going specifically for the mitochondrial H2O2. Mitochondria doesn't have catalase. So if you increase the activity of those antioxidant enzymes, that's a good shot. So they do these SOD mimetics. It's called SOD mimetics, or whatever mimetics. That it acts like the enzyme and boosts the activity. And now those enzymes have a high capacity to remove these radicals instead of catching the radical itself, which we cannot.
So most of the time when they made those kind of study, would they recommend to consume antioxidants and vitamins and stuff?
Sure, consume them, but
But it's like they don't measure like.
well, traditionally, I don't want to bash them too much. Traditionally, they did not measure those antioxidant enzyme activities. I suspect what happens is when it works, and it does work, that it's a signaling that it's blocking. Right? So it happened with resveratrol and sirtuins. So resveratrol from the red wine, and that's why I justify my red wine drinking. It doesn't act because it's an antioxidant. It acts because it activates the sirtuins. Right? Because the signaling will be quick enough to compete with all those other reactions that they're going on. So yeah. So a lot of antioxidants turn out to be not really antioxidants, but activate or deactivate signaling mechanisms. And not that way, that's really quick. So that way you have a chance. And I can, sirtuins with resveratrol, and I had another one in my mind that turned out to be I think even PMI 5011 that we did here with the botanicals, like it turns out to be activating insulin signaling. But it's not that it's scavenging any radical, because avocado was the other one, and then blueberry, you have to eat 20 kilograms a day. But that's not going to happen. But if blueberry has these anthocyanins in the skin that actually blocks an ADPH oxidase, well, now you blocked an ADPH oxidase, which is a ROS-generating enzyme. That way it will work. So I think that's where the focus should be. It's some sort of a signaling mechanism that those, like I just brought up the blueberry because I know about the anthocyanins, or the red grapes, or whatever. They do act because they block those things. But to compete with these reactions, there's just no way.
So it sounds like red wine over white wine.
Anything that justify, what was that, vitamin C, it just comes and goes out. So it's just no way, I can't see a way how that would scavenge anything, It will a little bit.
Interesting. Any other comments, questions? Thank you very much. So you've got, what division did the NIH by name?
EBK, NIDK,
NID.
Pathobiology of kidney disease,
Yeah, okay.
which I'm sitting there now as an ad hoc, so
Okay.
that will help hopefully, because they kind of pulled me in when I got a 20% off. They're like, ooh, this is really enough now, we're fully in now. We'll see who you are. No, I think people are heading that way, like what you asked, but a lot of studies in diabetes fail, and I just remember what you mean, because